Greetings! For today's video, we're going to discuss about the diseases of immunity. As for the members, I am Prilin Baguiwen. The others are Carlo Cabonita, Ethelyn Cañero, Jason Tabucol, and Decilon Valdez. With these diseases, it will going to affect the immune system of the body. So, let's define what is immune system. So, immune system is like a two-edged sword. On the one hand, organisms depend on the system for their survival. On the other, they fall prey to its many disorders that could threaten their very existence. These disorders range from immunodeficiency states to hypersensitivity reactions. In other words, from too little to too much activity on the part of the immune system. Immune system is also composed of two components designed to protect the body against extracellular and intracellular pathogens. First one is the humoral immunity, which is mediated by soluble antibody proteins. And the other one is the cellular immunity, which is mediated by lymphocytes. So let's move on to the cells of the immune system. So the lymphocytes. The lymphocytes are the most important cell of the immune system, and it arises from stem cells in the bone marrow. The organs that regulate the production and differentiation of lymphocytes are called the primary lymphoid organs, and include thymus in both mammals and birds, bursa of Fabricius only in birds, and bone marrow in mammals. After originating from stem cells, the lymphocytes follow two pathways. So the first is the thymus-derived lymphocytes, T cells or T lymphocytes, which those travel through the thymus both in mammals and birds and responsible for cellular or cell-mediated immunity. And the other one is the bursa-derived lymphocytes, B lymphocytes or B cells, which travel through the bursa of Fabricius in birds, that's bone marrow in mammals. So as for this figure, it shows here the two components of the immune system, which is the humoral immunity in the left side and the cellular immunity in the right side. As for the others under these components, we'll be going to tackle or discuss as we go along. So next, we'll be going to discuss the T lymphocytes. So T lymphocytes are essential for inducing the B-cell-derived humoral immunity. They can either directly lyse or destroy targets achieved by cytotoxic T cells or arrange for the antimicrobial immune response of other cells by producing soluble protein mediators called cytokines. In humans, T cells constitute between 60% and 70% of the lymphocytes in circulating blood, and in domestic animals, it is up to 80%. In the chicken, T cells constitute between 60% and 70% of the total lymphocyte population. So besides blood, T cells are also found mainly in the splenic periarterial sheaths and lymph node interfollicular zones. Each T cell is genetically programmed to recognize a specific process antigen or a peptide fragment by means of a T cell receptor, or the TCR. So the TCR is a protein heterodimer that is made up of two different kinds of polypeptide chains, which is the alpha and beta. So these two chains, the alpha and beta, are linked by a disulfide bond. So each chain has a variable antigen binding and the constant region. The TCR diversity for billions of potential antigens or the peptides is generated by somatic rearrangement of the genes that encode individual chains of the TCR. So the TCRs are non-covalently linked to a cluster of 5 constant polypeptide chains. These are composed of the gamma, delta, and epsilon proteins of the CD3 or the CD cluster of differentiation molecular complex and 2 zeta chains, as shown in this figure, the figure A. So, the CD3 proteins and zeta chains do not bind antigens or the antigenic peptides themselves, but they interact with the constant region of the TCR to transduce or transfer signals into the T cell after it has bound the antigen. So, with this figure, this is the schematic illustration of the TCR alpha and TCR beta polypeptide chains linked to CO3 and zeta chain molecular complex. So, in addition to these signaling proteins, the CD3 proteins and zeta chains, T cells express a variety of other constant function associated molecules including CD4 and CD8 as shown in this figure or the figure B. So with this, the TCR complex and its interaction with major histocompatibility complex or the MCH molecules on an antigen presenting cell. So the diagram represents the antigen recognition by CO4 plus T cells. So let's take note that the alpha-beta TCR heterodimer recognizes a peptide fragment of antigen bound to the MHC class II molecule. The CO4 molecule binds to non-polymorphic portion of the class II molecule. So the interaction between the TCR and the MHC bound antigen provides signal 1 for T cell activation. So the signal 2 is provided by the interaction of the CO28 molecule with the co-stimulatory molecules. CO80 or CO86 or the CD80 or CD86 expressed on antigen presenting cells. Similar interactions occur between MHC1 molecules and the TCR on CD8 plus T cells. In that case, the CD8 molecule binds to the non-polymorphic portion of the class I molecule. So the CD4 and the CD8 are expressed on 2 different types of T cells and serve as co-receptors for T cell stimulation. So the CD4 is expressed on about 60% of mature T cells known as helper T cells, whereas the CD28 is expressed on about 34% of T cells known as cytotoxic T cells. So in humans, in the normal healthy individuals, the CD4 or the CD8 ratio is therefore about 2:1. The CD4 negative and CD8 negative expressing T cells called CD4 plus, CD4 positive, and CD8 plus, CD8 positive, respectively, perform different but overlapping function. So the CD4 positive T cells are helper T cells because they secrete soluble molecules, the cytokines, that influence almost all other cells of the immune system. The CD8 positive T cells can also secrete cytokines, but they can play a more important role in directly killing virus-infected or tumor cells or the cytotoxic T cells. So, during antigen recognition, CD4 molecules on T cells bind to portions of class II MHC molecules on antigen-presenting cells, or the APCs. Likewise, the CD8 binds to class I MHC molecules. So, It is important to note that T cells require two signals for complete activation. So the first one is the signal 1, which it is provided by the interaction between TCR and MHC bound antigen. And the signal 2, which is it is provided by the interaction between CD28 molecules on T cells with the co-stimulatory CD80 or the CD86 molecules expressed on antigen-presenting cells. So the CD28 co-stimulation is essential because in the absence of signal 2, the T cells undergo apoptosis or became unreactive or anergic. So this two-signal arrangement may be a foolproof way to ensure that the normal individual successfully prevents autoreactivity. Because of the role played by the CD4 and CD8 coreceptors, the CD4 positive helper T cells respond to antigens only in the context of class II molecules, whereas CD8 positive cytotoxic T cells respond to antigens associated with only class I molecules. The CD4 T cells are of two types with different functions determined by their cytokines. First is the Th1 helper T1 or CD4 plus cells, which secrete cytokines that direct cell-mediated immune responses, including macrophage and natural killer cell activation. So these Th1 cytokines include interleukin or IL-2 and interferon gamma or the IFN gamma. So in contrast, we have the Th2 or the helper T2 CD4 positive cells, which secrete cytokines IL-4, IL-5, and IL-10 that antagonize the Th1 effects and or promote certain aspects of humoral immunity. Let's move on to the other one, which is the B lymphocytes. So B lymphocytes are the source of antibodies. So the antibodies participate in immunity either by directly neutralizing extracellular microbes or by activating complement and certain effector cells to kill microorganisms. They are present in the bone marrow, in peripheral lymphoid tissues, and in non-lymphoid organs such as the gastrointestinal tract. And in the lymph node, they are found in the cortex and lymphoid follicles and in the spleen in the white pulp. So B lymphocytes constitute 10% to 20% of the lymphocytes in human blood and 15% to 40% in domestic animals. In the chicken, these cells constitute between 30% and 40% of the total lymphocyte population. So, stimulation by local infection leads to the formation of a central zone of large activated B cells in follicles called a germinal center, which is a specialized microstructure that forms in secondary lymphoid tissues. So, after antigenic stimulation, the B cells form plasma cells that secrete globulins or the antibodies. So, immunoglobulins in turn mediate humoral immunity. So, of the 5 immunoglobulin isotypes, IgG, IgM, and the 19A constitute more than 95% of serum antibodies, whereas the IgE and IgD occur in traces. So each immunoglobulin has characteristics ability to activate complement or recruit inflammatory cells, and have defined roles. So for examples, the IgA is an important mediator of mucosal immunity, whereas the IgE has a special importance for helminthic infections and in allergic responses. So as for this figure, it shows here the immunoglobulin isotypes and also the B cell receptor complex and its interaction with antigen and co-stimulatory molecules. So with this, membrane-bound immunoglobulin, here shown as the IgM, binds to exogenous antigen. So the intracellular signal 1 for B cell activation is then provided by interaction with BCR-associated Ig alpha and Ig beta heterodimers. So the signal 2 is provided by activation complement interacting with B cell CD21 or by interaction of B cell surface CD40 with CD154 on activated T cells. So let's move on to the macrophages. So macrophages are part of the mononuclear phagocyte system, or the MPS. And it also plays important roles in the immune system. So the first important role of the macrophages in the immune response is macrophages, along with dendritic cells, express class II molecules. Macrophages therefore play the central role in the processing and presentation of antigen to CD4 positive helper T cells. So this is essential because T cells, unlike B cells, cannot be triggered by free antigen. Presentation of processed antigen by macrophages or other antigen-presenting cells, or the APCs, therefore is a must for induction of cell-mediated immunity. Next one is macrophages produce a large number of cytokines and are therefore important effector cells in certain forms of cell-mediated immunity. For example, delayed type hypersensitivity. So these cytokines not only influence T and B cell function, but also affect other cell types, including the endothelium and fibroblast. Next one, macrophages phagocytose and ultimately kill microbes coated by antibody and or complement. So therefore, they are important effector elements in humoral immunity. And last is macrophages kill tumor cells by producing reactive oxygen metabolites, or free radicals, or by secretion of tumor necrosis factor, or the TNF. Next is the dendritic cells. So cells with dendritic morphology, that is having free dendritic or branching like a tree cytoplasmic processes, occur in two functionally different types. First is the dendritic cells, or interdigitating dendritic cells. These are non-phagocytic cells and express highly of MHC class II and co-stimulatory molecules of the B7, CD80, and CD86. They are widely distributed and occur in lymphoid tissues and in the interstitium of many non-lymphoid organs such as the heart and lungs. So we have what we call the Langerhans cell, which are similar cells within the epidermis. So the distribution and surface molecule expression of dendritic cells make them best suited for presenting antigens to CD4 positive T cells. Next is the follicular dendritic cells. So these cells are present in the germinal centers of lymphoid follicles in the spleen and lymph nodes. So they possess Fc receptors for the IgG, that is, receptors that bind Fc portion, or the constant portion of antibodies. Therefore, they effectively trap antigen found to antibodies. Next one is the natural killers. So they are somewhat larger than small lymphocytes and contain abundant eosinophilic granules and can lyse or destroy a variety of tumor cells, virally infected cells, and some normal cells without prior sensitization. So both in human and domestic animals, they compromise 10% to 15% of peripheral blood lymphocytes. So NK cells do not express TCR and are CD3 negative. Instead, NK cells express two types of surface receptors that enable them to kill neoplastic or virus-infected cells. So the first type is the activating receptor. It recognizes certain unknown molecules on target cells. And the other type is called the killer inhibitor receptor, or the KIR, that inhibits natural killers' cytolysis, or destruction, by recognizing self-class 1 MHC molecules. So as for this figure, this shows the natural killer cell receptors and killing. So normal cells are not killed because inhibitory signals from MHC class 1 molecules override or dominate activating signals. So in tumor cells or virus-infected cells, reduced expression or alteration of MHC molecules interrupts the inhibitory signals, allowing activation of the natural killer cells and lysis of target cells. So in other words, the natural killer cell do not lyse healthy nucleated cells because they all express class 1 MHC molecules. So if virus infection or neoplastic transformation reduces normal MHC1 expression, inhibitory KRL signals are interrupted and lysis occur. So the NK cells are identified by the presence of two cell surface molecules, the CD16 and the CD56. So the CD16 is of particular importance. It is an Fc receptor for IgG and gives NK cells an extra ability to lyse IgG-opsonized target cells. So this process, known as antibody-dependent cell-mediated cytotoxicity, or the ADCC, is described under the type 2 hypersensitivity. So NK cells also secrete cytokines and are important source of interferon gamma, or the IFN gamma. So let's move on to the next slide, which is the cytokines, soluble mediators of the immune system. Cytokines are low molecular weight polypeptides, 10 to 40 kD, that are secreted by lymphocytes as well as by effector cells and the APCs. So cytokines mediated by short-acting soluble mediators, and they are so called because they are derived from cells. G, cytos, means cell. So with this, the induction and regulation of immune responses require the interactions of many cell types, including lymphocytes, monocytes, and other inflammatory cells and endothelium are properly organized and coordinated. So many interactions require cell-cell contact. So we have here the general properties of cytokines. First one is cytokines mediate their effects by binding to a specific high affinity receptors on target cells. For example, the interleukin-2 or the IL-2 activates T cell by binding to IL-2 receptors. So blockade of the IL-2R or the IL-2 receptors by a specific anti-receptor monoclonal antibodies prevents the T cell activation. So this is used to treat rejection of transplanted organ. Number 2, cytokines produce their effects in 3 ways. We have the first, the autocrine effect. In this, cytokines act on the same cell that produces them. Example, when IL-2 produced by activated T cell promotes T cell growth. Number 2 is the paracrine effect. In this, cytokines affect other cells in their vicinity. Example, when the IL-7 produced by marrow stromal cells promotes the differentiation of B-cell progenitors or the ancestors in the bone marrow. And the last one is the endocrine effect. So in this, cytokines affect cells systemically. So example of this is when IL-1 and the TNF that produce the acute phase response during inflammation. Number 3, cytokines can act in amplifying sequences. So for example, the TNF induces IL-1 production, which in turn drives IL-6 synthesis. Next, same cytokines are produced by different cell types. So for example, the IL-1 and TNF can be produced by virtually any cell. Next, number 5, the effects of cytokines are pleiotropic. That is, they act on many cell types, causing many different effects. So for example, the IL-2, initially described as a T cell growth factor. It regulates the growth and differentiation of B cells and the natural killer cells. Number 6 is many cytokines may produce similar effects. So that is, they are redundant or superfluous. For example, the IL-1 and TNF produce very similar effects. And last property of cytokines is that cytokines may be antagonistic. That is having opposing effects. In this way, they can finely regulate the intensity and type of an immune response. For example, interferon gamma activates macrophages, while the IL-1 or IL-10 prevents macrophage activation. So next is the general classes of cytokines. Cytokines can be divided into 5 categories on the basis of their general properties. First, cytokines that mediate innate immunity. So this group includes the IL-1, TNF, IL-6, and type 1 interferons. So interferons protect against viral infections, while IL-1, TNF, and IL-6 initiate non-specific pro-inflammatory responses, such as the activation of endothelium and mononuclear inflammatory cells, and induction of acute phase reactant synthesis by the liver. So macrophages are the major source of these cytokines. Number 2, cytokines that regulate lymphocyte growth, activation, and differentiation. So these are the IL-2, IL-4, IL-5, IL-12, IL-15, and the transforming growth factor alpha or the TGF-alpha. So some, such as the IL-2 and IL-4, favor lymphocyte growth and differentiation, while others, such as IL-10 and the TGF-alpha, downregulate or decrease the immune responses. So some of these cytokines, for example, the IL-2, fall under the group of the Th1 cytokines, which direct cell-mediated responses, whereas others, such as the IL-4 and the IL-10, are Th2 cytokines, which antagonize the Th1 effect and or promote certain aspects of humoral immunity. Next, number 3, cytokines that activate inflammatory cells. So, these include the interferon gamma, a Th1 cytokine, TNF lymphotoxin, TNF-beta, and migration inhibitory factor. So most of these cytokines are derived from T cells. Next, chemokines are cytokines that recruit inflammatory cells to site of injury. So these occur in two structurally different groups, CC and the CXC chemokines, based on the arrangement of certain cysteine residues. So the CXC chemokines, which include interleukin-8 or the IL-8, are produced by activated macrophages and tissue cells such as endothelium. The CC chemokines are produced largely by T cells and include monocyte, chemoattractant protein 1 or the MCP1, and monocyte inflammatory protein 1 or the MIP1 alpha. So number 5, cytokines that stimulate hematopoiesis. So many cytokines derived from lymphocyte or stromal cells stimulate the growth and production of new blood cells by acting on hematopoietic progenitor or the ancestor cells. So they are called colony stimulating factors or the CSF because they were originally identified by their ability to promote the growth of hematopoietic cells colonies from bone marrow precursors. So lymphocytes and bone marrow stromal cells are the major source of these cytokines. So examples are the granulocyte macrophage or the GM and CSF and granulocyte or the GCSF. So next will be the histocompatibility molecules. So for the general properties of histocompatibility. So these were first detected as the antigens that cause rejection of transplanted organs. Hence, the name histocompatibility, Greek or histo, meaning tissue, for example, the tissue compatibility. However, histocompatibility molecules are now known to play an extremely important role in inducing and regulating the normal immune functions. Remember, T cells, unlike B cells, recognize only membrane-bound antigens, and only in the context of self-histocompatibility molecules. So this phenomenon is referred to as MHC restriction. Thus, the main role of histocompatibility molecules is to bind peptide fragments of foreign protein or antigens for presentation to appropriate antigen-specific T cells. So, based on their chemical structure, tissue distribution, and function, MHC gene products are classified into 3 categories. So, first category is this class 1 MHC molecules. So, each of these molecules is a heterodimer that is composed of 2 different molecules, the alpha chain and beta microglobulin. So, the extracellular portion of the alpha chain contains a clef where foreign peptides bind to MHC molecules for presentation to CD8 positive T cells. In general, the MHC1 molecules bind to antigens derived from proteins synthesized within the cells. Example is the viral antigen. So because MHC1 molecules are present on virtually all nucleated cells, virally infected cells can be detected, analyzed, or destroyed by cytotoxic T cells. Next category is the class 2 MHC molecules. So the MHC2 molecules are also heterodimers, composed of two different molecules, the alpha and beta subunits. So, like MHC1, the extracellular portion of the MHC2 heterodimer contains a clef for binding antigens. However, the MHC2 molecules differ from MHC1 molecules in several ways. Unlike the MHC1 molecules, the tissue distribution of MHC2 expressing cells is quite restricted. So they are expressed mainly on antigen-presenting cells and B cells. In addition, other cell types such as vascular endothelial cells, fibroblasts, and renal tubular epithelial cells are induced to express the MHC2 by interferon alpha or the IFN alpha. In general, MHC molecules bind to antigens derived from proteins synthesized outside the cell. For example, those derived from extracellular bacteria. So this allows the CD4 plus positive T cells to recognize the presence of extracellular pathogens and to organize a protective cytokine-mediated response. And the last category is the class 3 proteins. So these include those complement components, the C2, C3, and the DF, that are encoded within the MHC. So genes for tumor necrosis factor, the TNF, and lymphotoxin, the TNF-beta, are also encoded within the MHC. Also, genetically linked to class 1 and class 2 antigens, class 3 molecules and the cytokines genes do not act as histocompatibility or transplantation antigens. So next will be the mechanisms of the immune responses. So the major function of the histocompatibility molecules or the MHC molecules is to present foreign antigens to T cells. Thus, histocompatibility molecules play an important role in the induction of humoral and cellular immunity. So first is the humoral immunity, or the antibody response, which has two key cells that involve in its development. So these are the B lymphocyte or the B cell, which produces antibodies to destroy an exogenous antigen. So an exogenous antigen is a foreign antigen that originates outside the body. For example, is the bacterial antigens. And the other one is the CD4 positive helper T cell, which regulates the immune response. So there are two types of exogenous antigens. These are the thymus-independent or the T-independent antigens and the thymus-dependent or T-dependent antigens. So B cells can bind both the antigens directly. That is, these antigens do not need any processing by the antigen-presenting cells. On the other hand, T cells require processed antigen. Thus, B cells differ from T cells in that they can bind free or unprocessed, whereas T cells can respond only to processed antigen presented in association with MHC molecules. Although B cells can recognize both the antigens directly, there's a basic difference between the two. So the T-independent antigens can produce antibodies without help from helper T cells. That is, in the absence of helper T cells. But they trigger only IgM response in B cells and can form IgG. This is because they fail to induce the formation of cytokines from helper T cells and therefore cannot trigger a class switch of antibody. T-dependent antigens, on the other hand, need the help of helper T cells. Without this help, they cannot form antibody. Since these antigens induce formation of cytokines from helper T cells, they can trigger a class switch and form the IgM, IgG, IgA, and the IgE. So the most antigens in the environment are T-dependent. That is, they require cytokine help from helper T cells to produce antibody.
So this is the mechanism of humoral immunity or antibody response. After entry, the exogenous antigen binds to B cell through cell receptors. These antigen are T-dependent and T-independent. If the antigen is T-independent, it causes B cells to generate antibodies without the assistance of helper T cells. And if the antigen is T-dependent, helper T cells required to provide cytokine support. In the T-independent, if B cells is triggered to produce antibodies, B cells could form plasma cells, which in turn produce antibodies. And if the antigen is T-dependent, helper T cells required to provide cytokine support. So, helper T cells, on the other hand, only bind and respond to processed antigen in the presence of MHC class II molecules. So, meaning, in the case of T-dependent antigens, antibody formation requires the coordination of two distinct mechanisms. First, the antigen should bind to B cells, and secondly, same antigen must also activate the helper T cells. So this is the flowchart of mechanism involved in the development of humoral and cellular immunity. Um, Cell-mediated immune response or cellular immunity. There are three types of cells that implicate in the development of cellular immunity. First is target cell, second is the helper T cells or Th1, and the third is cytotoxic T cell. So target cell, this can be antigen presenting cell that processes and expressing the endogenous antigen on its surface, and in this way gets to be the target cell for pulverization by the cytotoxic T cell. Th1 stimulate the immune response, and the cytotoxic T cell, this cell is referred as a serial killing because this is a destroyer of endogenous antigen in the target cell, then move to new target or infected cell. Mechanism of cellular immunity. To kill the target cell, cytotoxic T cells requires two signals. First signal occurs when cytotoxic T cells attaches to an endogenous antigen via receptors in conjunction with the MHC class I molecule. The second signal comes from helper T cells in the form of cytokines. And if this signal happen, of course, the target cell would be pulverized or destroyed. For Th1 cell to secrete cytokines, it requires two stimuli. First, it must respond to the same antigen as a cytotoxic T cell, but in the presence of MH class II molecules, and bind the antigen via its receptors. Interleukin 12, which is produced by the same antigen presenting cell that process of indigenous antigen, provides the second signal. The interleukin 12 act as a co-stimulator, interleukin 2 and interferon gamma, as well as IL-2 receptors, are secreted by the helper T cells. The second cytotoxic cell signal is interleukin 2 or IL-2. Th1 cells are the only ones that generate IL-2. IL-2 attaches to IL-2 receptors on the surface of cytotoxic T cells, causing them to generate additional IL-2 receptors in return. The interaction of these signals activates cytotoxic T cells, which divide and form a new population of cytotoxic T cells, resulting in the cell-mediated response. As a result, IL-2 is an important part of the immune response, and one of the important functions of cytotoxic T cells is to eliminate virus-infected cells from the body. Mechanism of immune-mediated injury or hypersensitivity reactions. Although immune responses are usually protective against infection and to some extent tumors, such response may also potentially damage host tissues. When humoral or cell-mediated immune responses to external or endogenous antigens generate tissue-damaging reactions. All such immune-mediated injuries are referred to as hypersensitivity reactions, and the resulting tissue lesions is called hypersensitivity disorders or illness. And sometimes, the term hypersensitivity is misleading. It is because it denotes an aberrant or excessive sensitivity to an antigen. But hypersensitivity illness can be caused by a perfectly normal immunological response to an antigens. And hypersensitivity diseases are best classified on the basis of the immunological mechanism initiating the disease. This method elucidates the mechanism through which immune responses is result in tissue harm or illness. The hypersensitivity reactions are classified into 4 types. 3 are variations of antibody-mediated injury, and whereas the 4th is cell-mediated. First is the type 1 hypersensitivity or the anaphylactic type. The mechanism of type 1 hypersensitivity results when IgE antibodies connect to certain antigens on mast cells or basophils. These IgE molecules are stimulated to release vasoactive amine and other mediators from mast cells or basophils, which impact vascular permeability and smooth muscle attraction in many organs. So later on, you want see the example of this different type of hypersensitivity. So for now is the mechanism only. And then the type 2 hypersensitivity or the antibody to fixed tissue antigen. Humorous antibodies bind to fixed tissue or cell surface antigens, causing the disease by predisposing cells to phagocytosis or complement-mediated lysis. This leads to an pathogenic condition. And type 3 hypersensitivity reactions are best known as immune complex disease. Antibodies attach to antigen, forming massive antigen body complexes that develop in blood vessel and activate complement. Neutrophils are drawn to immune complex and complement activation fragments. The tissue damage is completely caused by activated complement and the release of neutrophilic enzymes and other toxic chemicals. So that is the type 3 hypersensitivity, while the type 4 reaction or hypersensitivity reaction, or also called the delayed type of hypersensitivity, are cell-mediated immune responses. T cells or macrophages are activated as a result of cytokine release, resulting to tissue damage. So this is the table form of mechanism of hypersensitivity that I said earlier or recently. The type 1 hypersensitivity reaction or the anaphylactic type. The typical example of disease are anaphylaxis and some forms of bronchial asthma. The type 2 reaction is the typical example of this is autoimmune hemolytic anemia, transfusion reactions, hemolytic disease or of the newborn. The type 3 reaction, the typical example of disease is Artus reaction, serum sickness, certain forms of acute glomerulonephritis. And the type 4 or cell-mediated, the typical example of this is tuberculin reaction, tuberculosis, contact dermatitis, and transplant rejection. Type 1 hypersensitivity, anaphylactic type hypersensitivity, or immediate hypersensitivity, allergy, and anaphylaxis. This is a rapidly occurring reaction. It follows interaction of allergen with IgE antibody previously bound to the surface of muscles and vasculature in a sensitized host. In type 1 hypersensitivity, it can manifest as a local reaction or progress to autofatal, systemic, or anaphylaxis depending on how the allergen enters the body. The most significant cells in the development of type 1 hypersensitivity are mast cells and basophils. Mast cells are bone marrow-derived cells. They are present all over the body, but especially in the blood and subepithelial areas. Membrane-bound granules with a range of biologically active mediators are found in their cytoplasm. Basophils are similar to mast cells but are not present in tissues. Instead, they are circulate in tiny number in the blood and can be recruited to inflammatory areas like other granulocytes. Pathogenesis of type 1 hypersensitivity reaction. Type 1 Type 1 reaction are mediated by IgE antibodies. First, there has to be an exposure to certain allergens. The allergens stimulate in induction of CD4 plus T cells on the Th2 type. These CD4 plus cells play a critical role in the development of type 1 hypersensitivity because the cytokines emit drive B drive B cells to produce IgE and act as a growth factor for mast cells. In addition, eosinophils must be recruited and activated. IgE receptors bind to Fc receptors expressed on mast cells and basophils with high affinity. The individual is poised to acquire type 1 hypersensitivity once the most cells and basophils have been armed in this way. So, this is the image of the pathogenesis of the type 1 reaction. Primary mediators. These are performed, that is, they are already present within mast cells granules. After IgE triggering, primary mediators are released. So, primary mediators increase vascular permeability, vasodilation, bronchoconstriction, and mucus secretion are all caused by histamine, which is the histamine is the most important of these mediators. Adenosine, which promotes bronchoconstriction and inhibits platelet aggregation, and neutrophil and eosinophil chemotactic factors are two more rapidly released mediators. Heparin and neutral proteases are only a few of the mediators found in the granule matrix. The secondary mediators, this include two classes of components. First is lipid mediators and second is cytokines. The lipid mediators are generated by the activation of phospholipase A2 in cytokines, muscle-derived cytokines, TNF, IL-1, IL-4, and IL-5, and IL-6. The IL here is the interleukin. And chemokines play an important role in type 1 hypersensitivity reaction. See more in figure 68. So, this is the cross-linking of antigen in hypersensitivity 1. So, in flowchart, presented in flowchart, the activation of muscles following the cross-linking of IgE by antigen leads to release of their mediators. Clinical manifestation of type 1 reaction or may occur as a systemic disorder or as a local reaction. This depends on the route of antigen exposure. Systemic administration of protein antigens such as bee venom or drugs such as penicillin is result in systemic anaphylaxis. Local reaction occur when antigen is confined to a particular site because of the route of exposure, such as skin, gastrointestinal tract, or lung. Localized anaphylactic reaction in people include common skin and food allergies, hay fever, and asthma. The susceptibility is determined by genetics. Atopy is a term used to describe the familiar predisposition of such localized reactions. Type 2 hypersensitivity or the antibody dependent. Type 2 hypersensitivity is mediated by antibodies. These antibodies are directed against target antigens on the surface of cells or other tissue components. So, antigens might be natural substances found in cell membranes or the extracellular matrix, or foreign antigens like medication metabolites that are absorbed. And there are three different mechanisms of antibody-mediated injury in type 2 hypersensitivity. First is the complement-dependent reaction. Second, the antibody-independent cell-mediated cytotoxicity or injury or ADCC. And third is the antibody-mediated cellular dysfunction. The the complement-dependent reaction can mediate type 2 hypersensitivity in two ways: direct lysis and opsonization. In direct lysis, an antibody attached to a cell surface antigen promotes complement fixation and cell surface lysis via the membrane assault complex. In opsonization, cells covered with antibodies and complement C3b fragments and become prone to phagocytosis during opsonization. This method mostly causes harm to circulating blood cells due to the release of lysosomal enzymes and toxic byproducts. Antibody attached to non-phagocytosable tissue can cause frustrated phagocytosis and damage. ADCC, or the antibody-dependent cell-mediated cytotoxicity or injury. This type of antibody-mediated uh, can damage involves the death of cells that have Fc receptors for IgG. Without the phagocytosis or complement fixation, antibody-coated target cells are lyse. ADCC can involve a variety of leukocytes, including neutrophils, eosinophil-mediated parasite killing, and the utilization of IgE antibodies. And the third is the anti-mediated cellular dysfunction. These antibodies that can target cell surface receptors harm or disrupt the function without injuring of inflaming cells. Antibodies against acetylcholine receptors in the motor and plates of skeletal muscle, for example, disrupt of neuromuscular transmission in myasthenia gravis in humans, dogs, and cats, resulting to muscle weakness. Type 3 hypersensitivity of immune complex mediated. Type 3 hypersensitivity is mediated by the deposition of antibody complexes. This is followed by complement activation and accumulation of neutrophils. So, exogenous antigens like bacteria and viruses, as well as endogenous antigens like DNA, can be found in immune complexes. Many immune responses result in the formation of antigen-antibody complexes, which is normal antigen removal mechanism. When antigen is deposited, pathogenic immune complexes form in the circulation of extravascular location. Immune complex mediated injury can be systemic when complexes are formed in the circulation and are deposited in many organs or localized to particular organ, such as kidneys, joints, or skin. Both have the same mechanism of tissue damage. The sequence of events and situations that are contribute to the creation of immune complex, on the other hand, varied. Systemic immune complex disease. The, the pathogenesis of systemic immune complex can be divided into three phases. Phase 1 is the formation of antigen-antibody complex in circulation. Phase 2 is the deposition of the immune complex in various tissues initiating. And Phase 3, the inflammatory reaction in various sites throughout the body. So this is the three sequential phases in the induction of systemic type 3 complex, type 3 hypersensitivity. So, to explain the different phases, a typical example of systemic immune complex disease is a acute serum sickness. When significant amount of foreign serum were delivered for a passive vaccination, it was first observed in human. Specific antibodies are created about 5 days after foreign proteins are introduced. Antigen body complexes are formed when these react with the antigen that is still in circulation. That is the phase 1. In phase 2, in the second phase, the antigen-antibody complexes deposited in diverse tissue beds develop in the circulation. And in the third phase, deposition of the immune complex can cause a very localized activation of the complement cascade, resulting in generation of chemotactic peptides as well as the lytic unit. Um, there are two factors to decide whether immune complexes formation leads to tissue deposition and disease. First is the size of the immune complex, and second is the status of the mononuclear phagocytic system. So first is the size of the immune complex. if the immune complexes circulating in the blood are generally removed by macrophages. As a result of their overload malfunction, immune complexes remain in the circulation, increasing the risk of this tissue deposition. And in mononuclear phagocytic system, mononuclear phagocytic cell quickly remove very large complexes produced during antibody excess from the blood, making them generally harmless. The majority pathogenic complexes are small or intermediate in size and develop as a result of antigen overload.
So, type 4 hypersensitivity. Type 4 hypersensitivity is mediated by specifically sensitized T cells rather than antibodies. So, type 4 hypersensitivity is characterized by interactions between T cells and antigens that result in activation and cytokine production. So, it is of two basic types. First is the delayed type hypersensitivity and direct cell cytotoxicity. So, the delayed type hypersensitivity, THL type CD4 plus T cells secrete cytokines. These cytokines recruit other cells, especially macrophages, which are the major effector cells. That is, they induce the changes. So THL type CD4 plus T cells produce cytokines in delayed hypersensitivity. These cytokines attract other cells, particularly macrophages, which are the main effector cells or cells that cause changes. So the second is direct cell cytotoxicity. In contrast, in cell-mediated cytotoxicity, cytotoxic CD8 plus T cells themselves perform the effector function. So the effector function is performed by the cytotoxic CD8 plus T cells themselves. So let's proceed to the pathogenesis. So first, exposure of the animal to antigen tuberculin by injection or infection. So the first exposure of the animal to tubercle bacilli starts the DTH sequence of events. So this process results in the creation of DHL type sensitized CD4 plus cells that stay in the circulation for years. So the antigen is picked up by antigen presenting cells, macrophages, dendritic cells, process and in association with class 2 molecules presented to CD4 plus cells which recognize the antigen. So this leads to the formation of sensitized CD4 plus cells of the Th1 type. On second intradermal injection, memory cells respond to process antigen on antigen presenting cells and become activated, undergoing blast formation and proliferation. So activated CD4 plus T cells then secrete Th1 cytokines which cause delayed type hypersensitivity. So THL cytokines are secreted in tandem with this. It is these THL cytokines that are ultimately responsible for the DTH responses development. So let's proceed to the granulomatous inflammation. So granulomatous inflammation is a histologic pattern of tissue reaction which appears following cell injury. So infection, autoimmune, toxic, allergy, pharmacological, and neoplastic diseases are some of the causes of granulomatous inflammation. So the pathologic and clinical differential diagnosis as well as subsequent clinical therapy are narrowed by the tissue reaction pattern. So necrotizing granulomas, Non-necrotizing granulomas are all common reaction types. So mycobacterial infections are common examples of necrotizing granulomas. So within each category, however, there are broad differential diagnoses. So when a pattern-based algorithmic method is combined with clinical context, the cause can be identified. So the pulmonary system is one of the most prevalent places where granulomatous inflammation manifests itself. Granulomatous inflammation is a type of chronic inflammation that occurs in the body as a result of infection, autoimmune diseases, toxins, allergies, and even cancer. So the presence of mononuclear uh, leukocytes, specifically histiocytes, which respond to a variety of chemical mediators of cell injury. So let's proceed to the pathogenesis. So the origin of the epithelioid histiocytes begins within the bone marrow. When recruited into tissues, mature monocytes are renamed as histiocytes. So myeloid precursors grow into monocytes, which expand and enter peripheral circulation, giving rise to um, epithelioid histiocytes in the bone marrow. Then, when mature monocytes are recruited into tissues, they are termed histiocytes. So next is the activation of histiocytes via the innate immune response gives the cells their characteristic epithelioid appearance as compared to the inactivated reniform histiocytes with distinct cell borders. So next is within 24 to 48 hours of an injury, physiologic activation of histiocytes occurs via activation of complement helper T cells that release chemokines and cytokines to encourage, recruit, and drive macrophages to the site of injury. So, activated histiocytes persist beyond the acute phase and dominate the chronic inflammatory response and subsequent tissue repair. So, the existence of two types of histiocytes, one to balance the inflammatory effect and encourage phagocytosis, and the other to aid in wound repair, fibrosis, and to support by this persistent into the chronic phase. So let's proceed to the histologic subtypes. So granulomatous inflammation is commonly characterized by the formation of distinct granulomas composed of aggregates of epithelioid histiocytes. So granulomas form of aggregation of epithelioid histiocytes with a peripheral cuff of lymphocytes and plasma cells, and occasionally a necrotic center are common features of granulomatous inflammation. So the phrase granulomatous inflammation, on the other hand, refers to a wide range of observations from well-formed granulomas to loose aggregates of histiocytes mixed in with other inflammatory cells. So the pathologists use the latter to characterize the chronic damage and healing process or healing response, which is defined histologically as an admixed lymphohistiocytic inflammation with edema and early stage fibrosis. So this type of inflammation is non-specific, whereas identified granulomas can help diagnose underlying causes. So granulomatous entities can also be separated into organ systems that are typically affected, such as the lung, skin, kidney, liver, and lymph node. So this is how granulomatous inflammation looks like. So let's proceed to the etiology of granulomatous inflammation. So the etiology of granulomatous inflammation is broad and expands many etiologies, including infectious, autoimmune, toxic, allergic, and neoplastic entities. So next is the necrotizing granuloma. So microbacterial species are the most common etiologies of necrotizing granulomas worldwide. Chron- Chronic granulomatous response limits the extent of inflammation, activates histiocytes, and forms as rim of histiocytes and peripheral lymphocytes around a necrotic center. So let's proceed to the T-cell mediated rejection. It is mainly the result of cell-mediated immunity involving both delayed type hypersensitivity and cytotoxic T lymphocyte mechanism. So T-cell mediated rejection is defined by T-cell and macrophage infiltration of the interstitium, strong IFNG and TGFB effects, and epithelial degradation. A non-immunosuppressed host experience acute rejection in 10 to 14 days. So, cell-mediated immunity, which include both delayed type hypersensitivity and cytotoxic T lymphocyte mechanisms, is the main cause because the only significant antigenic difference between host and donor tissues is their histocompatibility molecules. Rejection is triggered by MHC. So there are two processes by which the host recognizes and responds to donor MHC. So the host recognition and response to donor MHC occurs by two mechanisms. First is indirect recognition and direct recognition. Indirect recognition, recipient CD4 plus T cells are stimulated by recipient APCs. So, indirect recognition occurs when recipients' CD4 plus T cells are activated by APCs that have acquired peptides derived from the donor's proteins, most commonly MHC molecules. Some of an allogenic graft's component proteins are shed into recipient tissues and picked up by recipients' APCs, or recipient APCs may penetrate the graft itself as cells die. Because most of these donor proteins are expressed by monomorphic genes within a species, peptides from these donor proteins are considered cells. So let's proceed to the direct recognition. Direct allorecognition is process by which donor-derived major histocompatibility complex MHC peptide complexes. So direct allorecognition is the method by which recipient T cells recognize donor-derived major histocompatibility complex MHC peptide complexes, which are normally presented by donor-derived um, passenger or dendritic cells. So let's proceed to the antibody-mediated rejection. So although T cells are of the greatest importance in the rejection of grafted organ, antibodies also play a vital role. Antigraft antibodies produced by sensitized B cells also contribute toward destruction of the graft through type 3 and 2 hypersensitivity reactions. So, after organ transplantation, antibody-mediated rejection is a common cause of graft loss. Anti-donor specific antibodies, particularly anti-HLA antibodies, cause it. CD4 was once thought to be a diagnostic sign for antibody-mediated rejection. Although T cells perform the most important role in the transplanted organ rejection, antibodies are also important. Through type 3 and 2 hypersensitivity reactions, Antigraft antibodies generated by sensitized B cells also contribute to graft death. The vascular endothelium is the primary target of antibody-mediated injury. So let's proceed to the autoimmune diseases. Autoimmune disease happens when the body's natural defense system can't tell the difference between your own cells and foreign cells, causing the body to mistakenly attack normal cells. So this here, when your body's natural defense system can't detect the difference between your own cells and foreign cells, it attacks normal cells, creating autoimmune disease. An autoimmune illness is a disorder in which your immune system assaults your body by mistake. Your immune system's job is to protect you from pathogens such as bacteria and viruses. It sends out an army of fighter cells to attack these foreign invaders when it detects them. So let's proceed to the immunological tolerance. So immune tolerance, also referred to as immunological tolerance or immunotolerance, is an active state of unresponsiveness to specific antigens in an effort to prevent destructive overreactivity of the immune system. It prevents an immune response to antigens produced by the body itself or recognized from a prior encounter. So immune tolerance, also known as immunological tolerance or immunotolerance, is an active condition of non-response to specific antigens in order to prevent the immune system from becoming overly reactive. It stops the immune system from reacting to active antigens produced by the body or recognized from a previous contact. So there are two types of immune tolerance, the self-tolerance and induced tolerance. So in self-tolerance, it refers to the ability of the immune system to recognize and therefore not respond against self-produced antigens. If the immune system loses this ability, the body can start to attack its own cells, which may cause an autoimmune disease. So, self-tolerance refers to the immune system's ability to recognize self-produced antigens and does not react to them. If the immune system loses its ability to do so, the body may begin to target its own cells, resulting in an autoimmune disease. So second is induced tolerance. So induced tolerance occurs when the immune system actively avoids responding to an external antigen. So when the immune system intentionally avoids responding to an external antigens, this is known as induced tolerance. So two major groups of mechanisms have been put forward to explain the tolerance state: the central tolerance and peripheral tolerance. So in central tolerance mechanism occur during lymphocyte development, either in the thymus for T cells or in the bone marrow for B cells. So for the peripheral tolerance mechanisms occur after mature lymphocytes are released into the lymph nodes or other tissues. So after mature lymphocytes are discharged into lymph nodes or other tissues, peripheral tolerance mechanisms kick in. These processes are designed to keep autoreactive immune cells from causing damage to the periphery if they survive the central tolerance mechanisms.
Mechanisms of autoimmune disease. Breakdown of one or more mechanisms of self-tolerance can result in an immunological attack on tissues and development of autoimmune diseases. Mechanism involved in breakdown of self-tolerance. Failure of activation induced cell death. Persistent activation of T cell may lead to their apoptosis through the fast ligand system. This means that defect in this pathway may allow persistence and proliferation of autoreactive T cell in the peripheral tissues. Number 2. Breakdown of T cell energy. Autoreactive cell that escape central deletion became anergic when they come in contact with self-antigen in the absence of co-stimulation. This means that such energy may be broken if normal cells that do not usually express co-stimulatory molecules can be inducted to do so. Number 3. Bypass of B cell requirement for T cell help. Many self-antigen have a large number of antigenic determinants. Some are recognized by B cell, other by T cell. Antibody response to such antigens occur only when self-reactive B cell receive help from T cells. Number 4. Failure of T cell mediated suppression. It reduces regulatory T cell function may result in autoimmunity. Number 5. Molecular mimicry. Some infectious agents share antigenic determinants with self-antigens. Therefore, an immune response against such microbes will produce similar responses to the cross-reacting self-antigens. For example, rheumatic heart disease in human sometimes follow streptococcal infection because antibodies to streptococcal M proteins cross-react with cardiac glycoproteins. Number 6. Polyclonal lymphocyte activation. Tolerance in some cases is maintained by energy of autoreactive T cell. Autoimmunity may occur if such self-reactive but anergic clones are stimulated by antigen-independent mechanism. The best example is bacteria lipoposaccharide, which induce mouse lymphocytes to form anti-DNA antithrombocyte and anti-red cell antibodies in vitro. Number 7. Release of sequestered antigens. Regardless of the exact mechanism by which self-tolerance is achieved, induction of tolerance requires interaction between a given antigens and the immune system. Number 8. Exposure of cryptic self-epitopes. It has been observed that molecular sequestration of antigens is much more common than is anatomic sequestration discussed under item number 7. From the many present, each cell protein has only a few antigenic determinants that are effectively processed and present to T cells. Genetic factor in autoimmunity. The precise role of MHC genes in autoimmunity is not entirely clear. Infection and autoimmunity. A variety of microbes, including bacteria, mycoplasmas, and viruses, have been shown to be involved in triggering autoimmunity. Autoimmune endocrine disease. Domestic animals suffer from autoimmune endocrine disease. They differ from human in that this tend to present a single organ disorder rather than involving many endocrine glands. Dog, human, and chicken suffer from a naturally occurring immune thyroiditis. In dog, it is associated with the presence of autoantibodies against thyroglobulin and thyroid colloid antigens. Autoimmune reproductive disease. These antigens are recognized by the adult immune system as foreign. A typical example of this occurs in male dog infected with Brucellia canis. This animal suffered from chronic epididymitis and became synthesized by sperm antigens carried to the circulation of phagocytosis by macrophages. Autoimmune ocular disease, an eye disease of horse known as equine recurrent uveitis or periodic ophthalmic, which can cause complete blindness, is believed to be of an autoimmune origin. Affected horse process circulating antibody against Leptospira interrogans. Autoimmune skin disease. This disease complex usually involve blister or vesicle formation in the skin. The complex consisting of 4 rare skin diseases that have been described in human, dog, horses, and cats. Pemphigus vulgaris, Pemphigus vegetans, Pemphigus foliaceus, and Pemphigus erythematus. Autoimmune hemolytic anemia. Autoantibodies to antigens on red blood cells cause their destruction and thus induce autoimmune hemolytic anemia. Hemolytic anemia are well recognized in human and dogs and have been recorded in cattle, horses, cats, and rabbits. Autoimmune hemolytic anemia in dogs are divided into 5 classes depending on the class of antibody involved. Class 1 AIHA. This is caused by autoantibodies that agglutinate red blood cells at body temperature. Class number 2 AIHA. This is usually mediated by IgM antibodies acting at body temperature. Class 3 AIHA. Most cases of AIHA in dogs and cats are mediated by IgG antibodies, which although can react with red cells at 37, cannot active complement or agglutinate the red cells. Class 4 AIHA. These antibodies are called agglutinists. Also, blood circulates through the extremities of animals. Class 5 AIHA. The fifth class of AIHA is usually mediated by IgM antibodies. These antibodies can only be identified by antiglobulin tests conducted in the cold. Autoimmune thrombocytopenia. Affected animal show many petechial in the skin, gingiva, other mucous membrane, and conjunctiva. These antibodies may also interfere with normal platelet function. The condition is usually observed as primary disease along with AIHA and systemic lupus erythematosus. It has been reported in horses, dogs, and cats. Epistaxis may occur and the dog may show melena and hematuria. Autoimmune muscle disease. Myasthenia gravis in human, dogs, and cats is a disease of skeletal muscle characterized by abnormal fatigue and weakness after even mild exercise. A dog with MC will collapse after trotting for only few yards. MC results for a failure of transmission of nerve impulses across the motor and plate of styroid muscle due to the deficiency of acetylcholine receptors. Discoid lupus erythematosus. This is a mild uncommon variant of SLE characterized by the occurrence of facial skin lesion. Sjogren's syndrome. In this syndrome, which occurs in both human and dogs, autoimmune attack in salivary and lacrimal glands leads to the conjunctival dryness and mouth dryness. Affected animal later develop gingivitis, dental caries, and excessive thirsts. Autoimmune polyarthritis. Most are associated with deposition of immunoglobulin or immune complex within joint tissues. 1. Erosive polyarthritis. Rheumatoid arthritis. This is commonest immune-mediated erosive polyarthritis. Rheumatoid arthritis is a common crippling disease in humans and is also seen in domestic animals, especially in dogs. Affected dogs may have depression, anorexia, pyrexia, and lameness. RA causes severe joint erosion and deformities. Pathogenesis. Rheumatoid arthritis commences as a sinusitis with extensive infiltration of lymphocytes in the synovia and neutrophils in the joint fluid. As the disease progresses, synovia swell and proliferate. Non-erosive polyarthritis. The joint cartilage is not eroded and the inflammatory lesion is largely confined to the joint capsule and synovia. Equine polyarthritis has been reported in the horse. Affected foals have many swollen joints involving all four limbs and persistent fever. Canine arthritis have been reported in the dogs. Feline polaritis occurs as chronic progressive arthritis of male cat. Dermatomyositis. It is a familial disease of dogs that resembles dermatomyositis in humans. It is mainly a clinical syndrome of dermatitis and less of myositis. Immune vasculitis. Several forms of immune vasculitis have been described in domestic animals. Their exact relationship are unclear, and as usual, they have been given several different names. These include canine juvenile polyarteritis in dog, polyarteritis nodosa in human, pigs, dogs, and cats, and leukocytoclastic vasculitis.
Now let us discuss the immunodeficiency diseases. These are disorders of the immune system that result in an immunological deficiency. It may be caused by primary immunodeficiencies, which are inherited defects affecting immune system development, and or by secondary immunodeficiencies, or the result from secondary effects of other diseases such as infection, malnutrition, aging, immunosuppression, autoimmunity, or chemotherapy. Animals with immunodeficiency show increased susceptibility to infections or parasitic agents and suffer from recurrent infections with pathogenic bacteria. Let us first discuss the primary immunodeficiencies. They are genetically determined and affect either specific immunity or non-specific immunity, mediated by complement proteins and cells such as the phagocytes or natural killer cells. First, we have the inherited defects in phagocytosis. Under it, we have the failure of opsonization, which has not been recorded in domestic animals except in canine C3 deficiency, which dogs show increased susceptibility to infections. And next, we have the failure of intracellular killing. Under, we have the Shigyaki-Higashi syndrome. It is a syndrome that can be seen in cattle, cats, rats, and humans. It shows an increased susceptibility to infection, a result from abnormally large granules in neutrophils, monocytes, and eosinophils, on which enlarged neutrophil granules result from the fusion of primary and secondary granules or abnormal lysosomes. Leukocytes are more fragile; they rupture spontaneously and have a defective chemotactic responsiveness and reduced mobility. The natural killer cells are also affected, thus the animals show increased susceptibility to tumors and some viruses. Next, we have the Pelger-Hewitt anomaly or the pH anomaly. It is a condition where the neutrophil nuclei fails to segment into lobes. The neutrophils are immature. It is observed in humans, dogs, cats, and rabbits. Next, we have the canine leukocyte adherence deficiency or the LAD. It is a condition where the neutrophils are not able to adhere to the endothelial cells due to the absence of integrins, thus it enables the invading bacteria grow freely and affected dogs suffer from recurrent infections and have large numbers of neutrophils in their bloodstream. Next deficiency, we have the bovine leukocyte adherence deficiency. It can infect calves on which they suffer from recurrent bacterial infections, delayed healing, and a persistent extreme neutrophil. Affected calves usually die between 2 and 7 months. Such calves have large number of intravascular neutrophils, but very few have extravascular neutrophils even in the presence of invading bacteria. Next, we have the inherited defects in the immune system. If both the cell-mediated and antibody-mediated immune responses are defective, the genetic lesion lies at a point before thymic and basal cell processing, that is, a stem cell lesion. Under the inherited defects in the immune system, we have the immunodeficiencies of the horses. It includes the SCID or the Severe Combined Immunodeficiency Syndrome that affects the foals. Affected foals fail to produce functional T or B cells and have very few circulating lymphocytes, on which they cannot produce antibodies and may result to agammaglobulinemic. It is a condition where the affected animals have no B cells and have a very low levels of all immunoglobulins. The affected fowls are born healthy but fall sick within 2 months. They all die by 4 to 6 months from severe infection by a variety of low-grade pathogens. On necropsy, the spleen lacks germinal centers and periarteriolar lymphoid sheaths. A thymus can be very difficult to find, and diagnosis should have first, an absence of circulating lymphocytes. Second, gross hypoplasia of the primary and secondary lymphoid organs. And third, an absence of immunoglobulin M from a pre-suckle serum. Next is we have the immunodeficiencies of the cattle. The severe combined immune deficiency in calf also includes selective Here in the immunodeficiencies of cattle, the affected calves are born healthy but may suffer from severe skin infections by 4 to 8 weeks and die later. There is atrophy of the thymus, spleen, and lymph nodes, and depletion of lymphocytes in the gut-associated lymphoid tissue or the gout. Next is the immunodeficiencies of dogs. An X-linked severe combined immunodeficiency or SOD in dogs characterized by increased susceptibility to infections and a lack of palpable lymph nodes. The affected dogs have low lymphocyte counts and major drop being in the CD8 T cell numbers. The puppies are hypoagammaglobulinemic with normal immunoglobulin M but lack in immunoglobulin G and immunoglobulin A. On necropsy, the thymus is about 10% of the normal weight. The lymph nodes are difficult to find. The spleen contain large periarteriolar lymphoid tissues with occasional small lymphocytes and have few plasma cells. The other immunodeficiencies of dogs include selective immunoglobulin deficiency, selective IgA immune deficiency, and transient hypoagglomeranemia, T cell deficiency, and acrodermatitis. And then last, we have the immunodeficiencies of chickens, which includes the selective immunoglobulin A and immunoglobulin G deficiencies. The immunoglobulin G deficiency is also known as the hereditary diamaglobulinemic. After the primary, we also have the secondary immunodeficiencies. The immune system is subject to destruction and dysfunction from infection by a variety of pathogenic agents. Most important are microorganisms, especially viruses and other agents, being toxins, stress, and malnutrition. First, we have the virus immunosuppression. Virus-induced immunosuppression. It can be divided into those that affect primary lymphoid tissues and those that affect secondary lymphoid tissues. The immunodeficiency syndrome of humans, a disease that results from infection with human immunodeficiency virus or the HIV, also similar to simian AIDS and in monkeys and feline AIDS. The canine distemper virus also destroys the secondary lymphoid organs. It destroys lymphocytes and produces lymphopenia. The canine distemper virus depresses the production of interleukin-1 and interleukin-2, while it also stimulates prostaglandin released by macrophages. As a result, the lymphocyte blastogenesis is depressed, immunoglobulin levels fall. The depletion of lymphoid tissues occurs in feline panleukopenia, canine parvovirus 2 infection, feline leukopenia, and African swine fever, where the virus localizes in germinal centers. The bovine virus diarrhea can cause lymphopenia and destruction both of the B and T cells. Infected cattle show the depressed neutrophil function. The herpes virus infections are also immunosuppressive. Next, we have the retrovirus infections in cats. We have here the feline leukemia. The feline leukemia virus is an oncogenic retrovirus, may cause different syndromes in cats. The virus first replicates in oral lymphoid tissue and infects all other lymphoid tissues. Cats have higher chance of lymphoid tumors and mostly are due to feline leukopenia virus, like lymphosarcoma. Immunosuppression of feline leukopenia virus It causes a destruction in lymphocytes and suppression of their function and the production of large amounts of immune complexes, leading to severe glomerulonephritis. Next, we have the feline leukemia virus AIDS. It is profoundly immunosuppressive and causes fatal nearly 100% of the infected cats. There is a drastic drop in CD4 T cells and shows a defect in inability to mount antibody responses. It occurs mainly in older male cats. It may cause lymphoproliferative immunodeficiency and neurological diseases. We have these stages of feline leukopenia virus AIDS. On the primary stage, the develop generalized lymphadenopathy and later on the leukopenia. Mostly, the cats can recover in this stage. On the secondary stage, the cats develop aplasia due to lymph node gradual hyperplasia and bone marrow suppression. On the third stage, the progressive generalized lymphadenopathy occurs. Then on the fourth stage, secondary bacterial infections are occurring. On the final stage of feline leukemia virus AIDS, they suffer severe age-like diseases, the opportunistic infections. They show immunological defects and also anemia, lymphopenia, neutropenia, and most critical stage, no cats have been seen to recover on this stage. For the immunosuppression of feline leukemia virus AIDS, the infected cats have lower number of neutrophils, lower T cells, and higher B cells. The infected cats shows major drop in the number of CD4 T cells. Next, we have the retrovirus infections in cattle. The bovine immunodeficiency virus, or the BIV, on the infected cows shows lymph node hyperplasia, lymphocytosis, CNS lesions, and weaknesses. And we also have the retrovirus infections in dogs. The canine immunodeficiency virus has been isolated from dogs. In necropsy, a dog shows depletion of lymphoid organs and bone marrow hypoplasia. Next, we have the other causes of secondary immunodeficiencies. First, we have the microbial and parasitic infections. Immunosuppression, the infestation with toxoplasma or trypanosomes, helminths, Trichinella spiralis, arthropods, demodex, and bacteria Pasteurella hemolytica, the actinobacilli, and some streptococci. Next, we have the toxin-induced immunosuppression. The environmental toxins such as the dieldrin, iodine, lead, cadmium, methyl, mercury, and DDT are immunosuppressive. Third, we have the malnutrition in the immune response. In general, severe nutritional deficiencies reduce T cell function and therefore impair cell-mediated responses. Starvation results in depressed complement levels and impairment of neutrophil and macrophage chemotaxis. The respiratory burst, release of lysosomal enzymes, and microbicidal activity. Fourth, we have the exercise and immune response. It increases immune function. Exercise raises blood neutrophil levels and increases the natural killer cell activities. However, strenuous exercise is stressful and increases susceptibility to infectious diseases. This is due to raised steroid levels due to stress. An intensive prolonged exercise may induce a functional deficiency. Fifth, we have a trauma and immune response. A severely injured animals die from a sepsis and as a result of immunodeficiency. And last, we have the age and the immune response. The immune response usually decreases with the advancing age. Now let us talk about the acquired immunodeficiency syndrome or the AIDS, caused by human immunodeficiency virus that prolong the lentivirus family. It is characterized by profound suppression leading to opportunistic infections, secondary neoplasms, and neurological manifestations. For the pathogenesis of AIDS, it has two targets, mainly the immune response and central nervous system. It affects the cell-mediated immunity resulting from infection and severe loss of CD4 helper T cells, and the CD4 molecule is high affinity receptor for HIV. It explains the selective tropism or the affinity of the virus for CD4 T cells and its ability to infect the macrophages and dendritic cells. The T cells in HIV infections, profound loss of CD4 T cells from blood. The productive infection of T cells is mechanism by which HIV causes CD4 T cell depletion. There is also a loss of memory of CD4 T helper cells. The monocytes or macrophages in HIV infection. The infection of monocytes and macrophages is also important in the pathogenesis of HIV. The infected macrophages are found in tissues and provides a vehicle for HIV to transport the different parts of the body, mainly the nervous system. During the infection, while the CD4 T cells are lost, the macrophages remain and the major site of viral replication. Next, we have the dendritic cells in HIV. The mucosal and follicular cells are also called the Langerhans cells, capture the virus and transport it to the regional lymph nodes where the CD4 T cells are infected. The B cells and other lymphocytes, the patients show severe abnormalities of B cell functions. The CD4 T cells plays the central role in the immunopathogenesis of AIDS, especially in the immune response. Thus, a major loss of this master cell also affects almost all of the other cells of the immune system and causes immunodeficiencies, decreased protection of the body from infection and or other viruses, and may result to acquired immunodeficiency syndrome.